Thank you. One of the things I'd ask you to consider is, are we human? That may sound like a really silly question, but let's take the facts. We're made up of human cells, and we're made up of microorganisms. The microorganisms are really somewhat alien to us, or are they? The reality of it is that microorganisms are pervasive throughout the entire environment. They were part of what terraformed the planet 4.2 billion years ago into the living, oxygen-rich, water-rich environment we have today. They evolved into every single life form, and microbiomes are actually everywhere. The reality of it is, whether it is ourselves, where we have many different microbiomes that are a part of our own body, from a hair microbiome to an ear microbiome to nose to eyes to mouth to skin, we are covered in microorganisms in a way we really had no idea. In fact, they've just recently identified a rather robust microbiome in the brain. So the microbiome are part of us and become part of us at a very young age. In fact, when we are born, we pick up the mother's microbiome during birth and during feeding. And as we live our lives, we pick up various other microorganisms to fill out our own microbiome. Now let's put this in a little perspective, because the question I started with, are we human or what, is a good question. Because one of the things we know is that the communication between our microbiome is very intense. So even though we have these foreign invaders on our body, they control more than we ever thought. We're learning that they do things like tell us when we're hungry, what we may want to eat, when we want to eat. In fact, recent studies have even shown potentially your own microbiome and a partner's microbiome might be communicating to actually bring you two together. The facts about the microbiome are hard to get your head around yet. Science is just starting to figure this out. But let's talk about the real statistics, and that is, for every one gene you have as a human, your human microbiome has 100 genes. So your genetic material that you walk around with is swamped by microorganisms. You're actually outnumbered in your body 40 trillion microorganism cells versus 30 trillion. A lot of people didn't know that. Now, put it even in a wilder perspective, is that those microorganisms have a mutation rate that's very high. So it changes constantly. In fact, in the next 12 hours, every human in here will have 120 billion mutations in their own microbiome. So where did drugs come from? The interesting part is, over the last 60, 70 years, most drugs have come from microorganisms, and all microorganisms come from microbiomes somewhere. And that's either directly, indirectly, or inspirationally, because science has showed us some chemical property that we like, and we take it into the lab and we modify it and then use it as a drug. So some of the first drugs we got that I'm sure everyone's aware of is something like penicillin. It was two microorganisms in battle in 1928 in a lab, sitting in a petri dish next to each other, and the researcher noticed that the mold that was producing a chemical kept the bacteria away. Ultimately, that was turned into the first real antibiotic and used extensively in the 1940s. Saved a lot of people, but it came from microorganisms. Other places drugs have come from are things like plants, other creatures. The reality of it is something like Taxol came from the bark of a Pacific yew tree. So as they were looking at the bark of the tree, they noticed that it could kill cancer. And it's an active cancer-killing agent even today. And the belief is it came from the tree. The reality is we're also learning about horizontal gene transfer. And we knew that genes could transfer from microorganisms to other organisms, animal organisms, rather easily. But now we even have examples of microorganisms doing horizontal gene transfer to even plants. So when you think about the essence of life and its ability to communicate and its ability to change, and then the capacity to be able to change so radically, so rapidly, what it really states is we're pretty stable because we don't go through those changes. We actually push back on those changes dramatically as an organism, no matter what that onslaught is. But that's also one of the reasons it's very difficult to actually find drugs. It's not an easy thing to do. So back 60, 70 years ago, in the golden age of pharmaceuticals, the way they found drugs was they'd go take samples of microorganisms or samples of dirt or samples of anything and see what they could grow. Well, the challenge was, even if I took a sample of the floor right here and put it in a culture plate and incubated it, only 1 out of 100 organisms that I would sample would be able to grow. Only 1 out of 100 would I even have a chance to study. But there's even a tougher equation to that. And that is just because I can grow that one doesn't mean I can learn a thing. So if I grow that up in a culture and I study what it produced, odds are it won't produce anything because it takes life's interactions to produce things. And that's one of the challenges. But what turns out to be a 1 in 5,000 chance of producing something has led to most drugs on the shelf. What this ended up doing is it challenged researchers to try to figure out how do we find more drugs, more cures, more opportunities. And one of the things that came along in the 60s was a massive chemistry kit known as combinatorial chemistry. And when you look at having a super incredible chemistry kit to mix up anything and everything you want, you'd think that if you could automate that and do things with that and create what was ultimately called combinatorial chemistry, you would have an opportunity to find all sorts of potential drugs. Well, no. Billions and billions of dollars over decades. There's only one drug on the shelf. Now, why is that? Well, if they'd ask a mathematician, with what we know today, obviously not what we knew then, what we know is that small molecules that are the most drug-like molecules themselves, that there's nearly an infinite assembling of these because of how the elements come together, how they orientate, how they spin, what charges they are. In fact, it's estimated that there is 1 and then 500 zeros possibility of small molecules. So even if you had the super duper combinatorial chemical capability to do 1 billion molecules per second, every second for the next 8 billion years, you'd only be at 1 and 34 zeros. So mathematically, it's going to be very tough to just randomly stumble upon it. But the technologies that were created are great technologies because we've learned how to take existing molecules we do know and make them better. Take inspiration or take something from nature, and that's important. So let's talk about genomics. Well, we all knew 15, 20 years ago that when we had the human genome decoded, we'd learn everything we ever needed to know. We'd know what drugs would work on each people. We would know exactly how to solve disease. Well, we've certainly learned a lot. But what happened is then came along an epigenome. And an epigenome is what we go through every day in living life. And what they found out is 2 identical twins in very little time whatsoever are hardly alike genomically. Their genetics changes. And that's the environmental factors. It's what you eat. It's potentially your own microbiome interacting with you. But then we also learned about the microbiome, and it's 100 times the genomics that we have. So essentially, the complexity of genomics has gone to a whole different level. And mathematically, to use just genomics to find solutions, it's not possible. To use genomics with inspiration of other technologies, what goes on with real life, absolutely it's possible. So where is the future source going to be? Well, it's going to be the source that has always worked the best. It's going to be from the microbiome. And in fact, in June of 2016, there was a recognition and a presidential announcement called the Microbiome Initiative. So there already is a scientific recognition that the microbiome is absolutely key to our knowledge about us as humans. And there is a number of approaches to get at what these unculturable microorganisms can potentially offer us. And that's one of the things that our company is actually doing as well. We're going and digging into the environment where we can grab a microbiome and study it. And study it to a point where we can find the chemistry that could potentially be a next cure. There's others looking at the unculturable microorganisms, and there's a lot of different strategies. But the reality of it is, it has a proven, reliable source. It's not easy. Nothing's ever simple. As complex as this is, it's going to remain tough. But one of the things that's happening is when you look at all the tools that have been developed in the last 50 years, we're now on the verge of being able to use microorganisms in a way that we never could 40, 50 years ago. The tools themselves, and genomics and synthetic chemistry or combinatorial chemistry, along with many other technologies, are leading us down that pathway to new cures, new treatments, and a better world. So there's one thing I will say to the crowd, and that is, if you have an opportunity to go to a grad party, high school or undergrad, and you've got a student moving forward into higher education, I would say take the opportunity to give them one word, and that one word is microbiome. Thank you very much.